Everything set up. So today we're going to talk more about, last lecture we talked about making biomolecules, the sources, trying to purify those with centrifugation or dialysis, more simpler methods. We're going to get into some specialized methods of chromatography and electrophoresis. And I will note that a big chunk of my PhD thesis was on chromatography. So I was using fluorescence microscopy, which we talked about earlier, to study chromatography. So I could probably talk about this for a long time. So I'll try not to go on long tangents on the side there, or if something isn't clear, please stop me, because since that was a topic, if I'm not defining terms or using terms that you aren't aware of, please stop and ask the question. But with the discussion of purification, I just wanted to bring up that I saw this news story yesterday, which really emphasizes the importance of purification and making sure that you're doing it the right way and you're having pure products. That this is a story from Bloomberg where Eli Lilly in the plant, I think it's in New Jersey, where they're using that to make one of the antibody cocktails, that the FDA flagged them because within the text of this story that they were using the wrong materials. I'm guessing some of the stationary phase materials or some of the solvents maybe, or reagents in the purification step. So then when they were trying to test this and use quality assurance, they were running into issues, and they were trying to, from my understanding, the story kind of hide over that and stuff, downplay the quality missteps here. But just mentioning purification relevant right now with trying to make, it's difficult to purify monoclonal antibody mixtures since monoclonal antibodies are large. They're produced in cells where it's like that hot mess of cell lysate and everything. So just pointing this out, that what we're talking about in class, relevant to a news story, purification is important. And another, if you want to see another example of understanding your data using analytical and biophysical techniques correctly. I don't know if any of you guys have read the book Bad Blood about the Theranos and Elizabeth Holmes, and I think they had a documentary afterwards on HBO, but that's another thing where it's with caring about statistics, doing analytical things, doing biophysical tools, it just drives you crazy. So it's a very interesting book and story there. So just a aside there of the relevance. Okay, so outlining what we'll be going over today. This includes, we're continuing once you have an analyte, one of our desired biomolecules, how do you isolate it? Purify it? Similar to what we talked about last lecture, but we're focusing today on chromatography. And by the end of the lecture, you should know the different forms or types. There's a lot of categorization of different chromatographic techniques, so we're going to go over that and the physical basis behind why they're working. And we're also going to go over some of the numbers behind it, like how do you quantify the performance of a chromatography run and the like. So again, like last lecture, I said this is a method for separation, but it can also be used analytically, I guess, as an instrument. We'll go over that. And if we want to lay out if you're using this for a quantitative method, if you're trying to detect how much of something is there, and if it's there, I guess this isn't as similar to the spectroscopy, but I would say the source would be you inject your solution. And your solution is where your sample is, but the key thing is you're trying to separate it. So I guess maybe we can say like method to detect the sample. This would be like passing through column, where the column is the key aspect of the chromatography instrument. And then if you want to detect your sample, there's many different detectors that can be used. You can use UV vis, fluorescence, mass spec, which we'll discuss next week, NMR, like a lot of these different instruments and modes of detecting molecules can be hooked up to a chromatography instrument. And so this would be if you're doing like the analytical detection and more on the method side of things. After you detect what's there, you can then collect your product, your analyte, as an additional step there. So that's laying out how the instrument works a bit. So I do want to just remind you guys, we're talking about separation. So we did talk about this last lecture, that this slide is straight from last lecture, of that we're trying to isolate desired analyte by taking advantage of different forces or physical differences between what you want and everything else in the mixture there. So we talked about centrifugation and dialysis last week. And then today we're going to be talking about all of these other guys, taking advantage of charge, polarity, hydrophobicity, specific binding, and the like. So we're all these different terms in front of the chromatography term are mentioning the physical property that you're taking advantage of in the separation. So how does chromatography differ from these other types of techniques? Well, they're techniques using a column and some material. So we have to understand what some of the terms are used in the chromatography field. So chromatography gets its name from the fact that it was used and developed to separate chlorophyll in plant samples, which led to having separations with different colors. So the chroma, I guess just the chroma portion here means colors. And you can see here with some separating of, I think this is probably like food coloring or different dyes here, that you can see that some chromatography, you'll see these nice, beautiful, colorful bands. And that's where the name comes from. But chromatography, what you're doing is you're having your analyte in a solution, and that solution is called the mobile phase. And the mobile phase is passing over a solid. And that solid is called the stationary phase. And then your analyte versus other molecules have a different, let's say, number and time of interaction with the stationary phase. And that leads to the separation. So typically these stationary phase particles are packed in a column, and they're going to be beads of glass, so silica, or a hydrogel. And they're probably around 1 to 10 micrometers in size. So you can see this is a simulation here of all these blue beads are going to be your packed stationary phase in a column. And you have molecules passing through, binding, unbinding, diffusing between the particles. And you can see that there's a blue trace here, a green trace, a red trace. And when, let's say this is a protein, it's spending some time in that mobile phase diffusing around, it comes to the stationary phase particle, binds and then unbinds and leaves. So the difference between something not interacting at all, interacting intermittently, or having strong interactions leads to these separations. So you can see like for these blue, green, and red trajectories in the simulation, you can see over time and over the length of the column that will lead to the separations there. And also worth noting, I forgot to include this, that with the stationary phase particles with the silica or the hydrogel and their sizes here, these can also be porous. In the case of the hydrogel or the silica, they can be completely solid or they can be semi-porous. And all the aspects of the size and the porosity will lead to different numbers of interactions and different pressures that you can run your chromatography method there. And hydrogels and silica are used because they're you can take advantage of chemistry to then easily modify the surfaces of them, use chemistry. And silica is rather inert that then you can change your stationary phase for the separation that you need. So this type of terminology of mobile phase, stationary phase, that's going to be used with all forms of chromatography. There's also, we talked about all the different forms of chromatography based on the forces, and we're going to go into the detail with those. There's also more categorization based on the instrumentation, not just those forces I was talking about will describe the stationary phase that you're using, but you can also have, has to do with how the instrumentation is set up. So the most simple form of chromatography would be column chromatography, or also called like gravity-based chromatography, where you'll see these in labs where you have your column and it's just like set up on a ring stand and you're using the force of gravity for the solution to pass through. So these are for rather simple separations, but if you're trying to separate complex mixtures or need high efficiency, there's also high performance liquid chromatography or ultra high performance. Where a lot of terminology in chromatography comes from just historical terms. So with high performance liquid chromatography, you have your mobile phase under, there's high pressure. So this performed better than just gravity-based chromatography, so hence the term high performance. Then they developed methods to work at even higher pressure, so they called it ultra high performance liquid chromatography. But I would say this is one of the most common forms that are used, and the fact that we have the term liquid here has to do with what your mobile phase is. And we're going to be focusing pretty much exclusively on liquid chromatography. There's also FPLC, and this is fast protein liquid chromatography. And I would say, again, we're using liquid. The fast protein just means that this is instrumentation has been designed and modified specifically for protein separations, but it has similar concepts of HPLC where you're using high pressures and the like. We're not going to talk too much about gas chromatography, but this would be working with gas as your mobile phase instead of liquids. With biological, biophysical separations, this isn't used as much since biology is mostly in the liquid phase. And then also worth noting that there's SFC, I think it's SCF, supercritical fluid chromatography. Okay, it is SFC. And this is where a supercritical fluid is your mobile phase. So this is typically liquid CO2 is used for your mobile phase. And this can be used to speed up separations as well. So just be aware of these guys. We're not going to talk about them in much detail. What we'll be discussing more are all stationary phases that are used in forms for HPLC, gravity chromatography, FPLC, and the like. As I mentioned on the outline slide, chromatography can be used as a preparative method. So what I mean by that is like separating your analyte to use it. So that was more related to what we were discussing on Tuesday, like how can you get, after you make a protein, after you make some nucleic acids, how can you get that desired molecule out from everything else so then you can use it for an application. Versus it can also be used as an analytical method where you're separating to detect, quantify, see what's in your sample. So be aware that if you see chromatography in the literature, are they using it to quantify something versus like, oh, this is just a step so we could isolate something. So with this analytical method, you can get things about like the concentration, like the mass or the charge, etc. These properties will be depending on like what type of stationary phase that you use. So most chromatography instrumentation, if we're focusing on HPLC or FPLC, is a basic block diagram here. What you're going to start with is having some solvents, a solvent reservoir. And this can be one type of solvent or multiple. So the solvent reservoir is going to be your mobile phase that's going to be passing over your stationary phase. So if you have a constant composition, that's going to be called an isocratic separation. If it changes over the course of your separation, that's going to be called a gradient separation. So for a gradient separation, why would you be mixing different mobile phases? You can have conditions where you want to load your sample, where you're going to be using a mobile phase where you're ensuring that your analyte will have a strong interaction with your stationary phase, and then everything passes through. And then if you want to then kick that analyte off of the stationary phase, you'll want to use a different solvent there. So you can change and modify and have different mixtures of your solvents there. These solvents, to actually get into the instrumentation, have to pass through a pump. And these are traditionally reciprocating pumps. That have some ball check valves. It's going to be a well-designed pump, so you're not having bursts of solvents that would then change the flow rates. So that's a big component of the instrumentation to ensure that you're not having flow rates disrupt your separation. So this pump is then going to send your mobile phase into the workhorse of the chromatography instrument, the column. These will be, you commercially obtain these, and they can be around 5 to 30 centimeters in length, 4 to 10 millimeters in diameter. And I would say that's what you would see in a research lab-based setting. If you go to industry, these columns can take up like an entire portion of a room. They can be like 6 feet tall, they can be 3 feet in diameter or so, but on the lab-based scale, these are typical sizes that you see, and the length and diameter will determine how much material that you can separate versus and the loading capacity and how much you can pass through there, and also the lifetime of those columns. So you'll start this out by first just like saturating with the solvent. But you also have to have your sample go up. And to do that, you have an injection port where you inject your sample here. And ensuring when you're loading, this is so you can load your sample. It's traditionally, you can do this with like a syringe and a needle yourself. There's also nice designs where if you have a large volume of solution, it's actually an additional pump itself. Here, and then after you inject that, that mobile phase is going to pass it over the stationary phase. You're going to see, okay, how's that separation progressing? You'll have your detector. Like I mentioned, this can be UV vis, a fluorimeter. It can be based on charge or current that can pass through. With the ampermetric detection, can be a refractive index, even light scattering. So a lot of the different optical methods that we've mentioned, and more complex would be like a mass spec or NMR hooked up to that. And after detecting it, you can then send, you have the option, I guess I'll draw it this way. You can send whatever passed through the detector. It can either go to waste, it's something it doesn't have your analyte, or you can have a collector. So if you want to use this for a preparative method and collect the analyte after it passes through the detector and you know where it is, then you can collect this and then use it afterwards. Okay, so we're now going to get into like the specific stationary phases that are used. Are there any questions so far? Okay. Moving on, getting into that terminology, I guess, from the third slide or so. Getting into these different forces, the first ones we're going to talk about are called partition chromatography. This is going to be based on polarity of your analyte. I guess there's 3 types of partition chromatography. And the terminology, again, I mentioned that like a lot of this comes from history of chromatography, of when things were developed. So the first one is normal phase. So that's what this guy is abbreviated here. Normal phase chromatography. It was developed first. So it was the normal method of using partition chromatography. And this is where you have a polar stationary phase versus a non-polar mobile phase. So you can see here some common normal phase, stationary phases where you have carboxylic acids or aldehydes, amines, cyanate groups, and something that you would use normal phase chromatography if you have a polar analyte. So your polar analyte would interact preferentially with the stationary phase versus whatever would be nonpolar would stay in the mobile phase. This is in contrast to reverse phase. That's why it's called RP here. And despite these names of normal and reverse phase is actually used more frequently than normal phase chromatography. And this is the opposite of normal phase, where you have a nonpolar stationary phase and a polar mobile phase. So this would be if you have a nonpolar analyte, you have something like lipids, you can see that just like the hydrocarbon tails in lipids, you have similar things on reverse phase stationary phases here. So I'd say the most common one, I didn't count these carbons here. I'm guessing this would be like a C18 stationary phase. When you see in the literature people using reverse phase chromatography, it's going to be like C18, C8, or I guess this would be like a C4, where that is the length of the hydrocarbon chain that's immobilized on your stationary phase. So these are usually carried out on a silica stationary phase bead that then you chemically modify to have your phase from that silica surface. So I'd say most commonly these are used on silica particles that are solid. Sometimes they're porous silica particles, but the important thing is knowing what chemical groups are on there. So just starting this guy because it's used more frequently. Another form of partition chromatography that I guess you can say is based on what's the difference between polarity and hydrophobicity would be hydrophobic interaction chromatography. Or HIC. And then this is a variation of reverse phase that works with aqueous solutions. So reverse phase chromatography doesn't always work when you're trying to separate proteins because you have to have, they won't interact with this nonpolar stationary phase. It's too hydrophobic. So this is a little bit less harsh or less, I guess, hydrophobic than the reverse phase separations. So I guess this is like a very basic cartoon where it's not defining what the hydrophobic surface is. You can see that the protein is in water, and based on the ordering of water, if you have a hydrophobic core or aspects of the surface of the protein, it will preferentially interact with that. And you can drive the proteins to either interact with the surface or not based on salting in and salting out with the Hofmeister series. So the Hofmeister series has to do with, I have it listed here, and it has to do with how much, how water orders itself around the hydrophobic aspects of protein, where this was, Hofmeister found how this interacts with proteins, these salt ions. So by using specific concentrations of anions or cations, that would change the ordering of the water and drive proteins to the surface or not. Also worth mentioning is there's hydrophilic interaction chromatography as well, which I'm guessing is a form of normal phase designed for proteins. But there's so many terms in there, but we won't get into detail of that. So this is based on polarity. Also, I think most conceptually easy way of viewing chromatography is based on size. So size-exclusion chromatography, or SEC, and this has to do with the porosity of the stationary phase. So if I'm explaining chromatography to a non-scientist or just a general public, I use the analogy of like if you're making pasta and you pour your pot of water and your pasta through a colander, that's like a form of, I guess, size exclusion chromatography, where it's like water and your pasta there, if those are your two components. So similarly, size exclusion has to do with if you're working with a porous particle. This is like a really rough cartoon. This is your particle. Small things will be able to interact with the pores, while large things are excluded, hence the term exclusion here. So if you're looking at your chromatogram where you have like amount of analyte versus time, here if we call this big guy A, I guess we have A, B, C, and D here. The first peak that you're going to see elute out is going to be A, because it doesn't interact with the inside of these particles. Then you're going to see your other guys pass out here based on the size. So large things elute faster. And it's based on selective permeation. So similar to dialysis, where you have that selectively permeable membrane in dialysis, here with a single cutoff value, here you'll have multiple cutoff values, because these pores within the stationary phase particle also aren't going to be uniform. So you'll have there. There's also other forms or names for size exclusion based methods. So size exclusion chromatography would be something that you would have in a column for like HPLC or FPLC. There's also gravity based methods. And if you're using aqueous versus non-aqueous mobile phases, you'll hear this referred to as gel permeation or gel filtration, where all of this is still based on size exclusion. So you can see here for a gravity-based method, there's like gel filtration aspects or terminology that's used. And also in the polymer science field, you'll hear gel permeation chromatography much more frequently than you'll see size exclusion terminology, but they're all based on the same physical aspect of size exclusion, but the terminology can vary. Moving on to doing separations based on charge would be ion exchange chromatography. And here, this would be a form of adsorption chromatography. So the fact, and also when we get to affinity chromatography, that's adsorption. So you're really seeing an adsorption process as opposed to partition chromatography. There might not be like specific sites where your analyte is binding. It's just having preferential partitioning in one phase versus the other. It might diffuse along for reverse phase or normal phase. It might just diffuse or preferentially partition within that stationary phase. But here there's actually specific sites where your analyte is going to bond. So at the surface, okay, this is in the column, you'll actually have like specific ligands. Let's say this is positively charged, or if you have a negatively charged protein, it will actually come and bind to that ligand site, hence the name adsorption chromatography. So yeah, you have charged ligands on stationary phase. I'd say here the stationary phase is more the gel particles, the hydrogels, versus normal phase and reverse phase is going to be more of the silica type particles. So these are sold under names like Cepharos is like a trade name from Sigma, which means that it's an agrostationary phase. So with these charge ligands on the stationary phase, this is all going to be driven by electrostatics, where if you have, there's two forms, there's going to be cation exchange. And that means that you have a negative stationary phase. Negative stationary phase and positively charged analytes or ions that interact with it. So it's called ion exchange because at this, let's see, this is cation exchange right here. You have a negative SO3 group, sulfate group here. And depending on the pH, you'll either have like a positively charged ion, like salt. If you have salt in your solution, some sodium would be absorbed to the sulfate group here. And then the exchange process is that, like, if you have a lysine group, it will exchange with that sodium ion, and hence the name ion exchange here. And then based on the pH, if you're doing a gradient elution, then you can tune when you want your protein absorbed versus exchanging it for the salt ions. Let's say you want to retain versus elute based on your pH there. So there's cation exchange. There's also anion exchange, which would be the opposite, where you have a positive stationary phase and a negative analytes exchanging with a negative ion there. So the positive stationary phases, I would say, are more based on amine groups. The cation exchanges, you'll have carboxylic acids or the sulfates for your stationary phases. And yeah, I mentioned you can use pH. You can also use like salt concentrations to then elute, control between retaining or eluting your analytes there. So there's other forms of, so ion exchange chromatography, that's one way to take advantage of electrostatics. Another way is based on charge is doing electrophoresis. So if you have a potential difference, you can also drive separations. So in the separation, I'd say for analytical methods of trying to detect what's there, there's capillary electrophoresis. Which is shown here, where you have cation and anionic solutions and you have a potential. And this orange guy here is going to be your capillary. With this potential, you're going to be taking advantage of electroosmotic flow. We're more positive. You can see the zoomed in version here. Based on that potential, more positive charges will flow faster versus negative charges. It's worth noting this capillary is just a thin glass tube. There's no stationary phase. Hence why it's not a chromatography technique. It's separate from that because there's no traditional stationary phase there. There's just a glass surface. And when you would pick to use capillary electrophoresis versus ion exchange chromatography is capillary electrophoresis is much more sensitive. So you can get higher resolutions than ion exchange. So if things are only different by a single charge, this is when you would want to be using capillary electrophoresis. I'd say more on the preparative side of things, electrophoresis in biophysics, you'll see used as gel electrophoresis. And this would be like the final step after when we talked about protein expression, or let's say you're also making DNA. That's when you'll use gel electrophoresis to determine if something is there or not, and also to separate it out at sometimes. So DNA. And gel electrophoresis does this detection entirely based on the molecular weight. In the form of, for proteins, this would be SDS page, which stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis. I'm sighing because it's a very long name and everybody just calls it SDS page. So you can see SDS page here. So for SDS page, what this means, SDS, the sodium dodecyl sulfate, is a soapy substance that you add to your protein solution that completely denatures the protein. So all of your proteins are unfolded and then you pass that through the gel. So the polyacrylamide here has size-exclusion properties because it's porous. I guess the polyacrylamide gel. So the SDS denatures. You pass it through the size-exclusion gel, and the electrophoresis means that with this potential here, you're driving the separation through the gel here. So applying a potential. So this here, you'd have different samples in your well. This is your gel. There's going to be some electrodes at the top, and also in the bottom here, there's going to be an electrode. So you can see the electrodes here. So as you apply that potential, things are going to pass through this gel specifically based on their molecular weight because they are completely denatured. If they were folded, there would be aspects of the size included in there and of the conformation of the protein. Because they're completely denatured, it will be only separated based on the molecular weight. So larger proteins will stay at the top, smaller ones will be at the bottom, and you can determine, okay, how many proteins are there if you know the molecular weight. I think this guy is based on DNA, 1,000 base pairs. Let's say you're looking for a DNA plasmid you made. Within a cell, you want to understand if that cell took it. You can find this band and determine, okay, is it there or not? So protein gel electrophoresis is done vertically. For DNA gel electrophoresis, that's usually done horizontally based on the different gels and processes there. Okay. Any questions so far? Let me just pause. Two more slides and then we'll get to the breakout session, just to tell you guys where we're at. Okay, last forces that I want to mention that are used in chromatography would be, the most specific form would be affinity chromatography. So this is where you have very specific recognition. It's on the stationary phase. So you can see two forms illustrated here, where you can have like a receptor ligand. So then you can have like all your other proteins or biomolecules in solution that would not fit in with this specific receptor. If we want to use like the somewhat incorrect terminology or contested terminology of the lock and key type mechanism, that's what you could take advantage of here. Or there's also metal-based, or it's chelation-based, where you can see that this will be something like a nickel NTA column, where metal ions nickel will coordinate with histidine residues on proteins. So if you mutate your protein to have a histidine tag, you can use these type of affinity columns to then separate that. This is commonly used in protein expression. So the nice thing with affinity chromatography is you have high selectivity for your specific analytes. But some drawbacks are, okay, the costs, the lifetime of your column, and yeah, I'd say, and generalizability, because with affinity chromatography, you'll be wondering, like, why don't you just use affinity chromatography for every single type of separation? If you have a column that has been designed, let's say this receptor ligand designed for your analyte, that's great. But if you don't know a receptor that will bind your biomolecule, let's say in like drug development, that receptor might not be known. You might have to actually design that receptor. So that's where like the cost, and it's not general enough there. So this can be used, I'd say, on like the lab scale for protein expression and the like. But the reason that you won't see that used as a common method, let's say in industry, is you might not have those aspects there. There's also challenges in removing the analyte after it's bound. So how can, if this is such a strong interaction, how do you break it off? How do you introduce something that would compete and kick off your analyte afterwards? So with like nickel NTA columns with histidines, you can load in a bunch of imidazole, which has a similar chemical structure, then kick off your protein in those type of separations. So we'll see this used sometimes, but not as general. Not for this, I would say this is much more for like the preparative methods, but you won't see affinity chromatography used for analytical techniques where you're determining like how much of different components are there. So those are the different forms of chromatography. There's other ones that I didn't discuss. There's chiral chromatography, thin layer for anyone who's taken organic chemistry, ion pair, supercritical fluid chromatography we didn't talk about. And there's also multidimensional, let's say multimodal, so where you're combining forces. So you can have something that would be, let's say, ion exchange and hydrophobic chromatography combined on a single stationary phase. And you can also have multidimensional chromatography, where you have a column that actually has 2 dimensions, and based on different forces in each dimension that you can lead to separations. So that's an expanding, focus on analytical techniques there. So realize there's a whole wide range of stuff with chromatography that we could discuss even more. Okay. So last thing I want to get at is a biophysics class, there's physics, so we have to have some numbers here as well. And as a disclaimer, this is the last point that I have on my notes, but I want to say it up front that all of the quantification is like empirically based. So it's kind of like, can you assign some numbers to talk about the performance? But those numbers have little connection to the physical phenomenon behind what's going on in chromatography. And that's an area of interest in my research there. Like, can we get more numbers more connected to like the physical chemical like phenomenon that's happening in the column? So just as a disclaimer, like I kind of hate some of these terms that they use because I think there could be a lot more physics in here. And I would say also these numbers haven't really changed since like the 1950s, that I think there can be more research done there as well. So some very general terms, okay, some of these numbers make some sense, is mobile phase time, which we'll call TM. How long is your analyte? How long would something that's not interacting with the stationary phase in the column? So I guess that would be this guy here. You might add some internal standard that you know would not interact with the stationary phase and see how fast that passes through. So that's mobile phase time. Then there's retention time. TR, and that's where your peak is located at here. So TR, so how long, the time it takes for your analyte to go through. Okay. So if you want to adjust adjusted retention time, that would be TR prime. That's going to be adjusting your retention time for like the mobile phase time. So that would just be simply TR minus TM. So everything is adjusted. That's why I said the mobile phase time is going to be based on an internal standard. So adjust all your peaks based on that internal standard. There's capacity factor. So relating, normalizing, I guess, your retention time. This is K. So you'd have your TR prime over your mobile phase time. So that's a normalization, again, to have all of your, I guess this is K prime, lowercase K prime. Oh, that's a way to compare different peaks here, your retention times relative to one another with that capacity factor. So we have, let's say we have two components here, TRA, TRB, two different analytes here. If you're trying to assess how the resolution, how well you're separating these things and compare them, there's also relative retention. So that's a ratio of their time in the stationary phase, and this is given the variable alpha, and that's just going to be your relative retention time of A over the relative retention time of B. So getting a ratio, while resolution will have to do with like the peak width as well. So this peak width we're going to call W. So the resolution would be the difference between those two peaks, their positions, divided by their average width there. So how well you can resolve those, because you can imagine if these peaks expand out, they're going to start to overlap, so you need some term for the width there. I have an example here of some separations that also, this peak width is also missing any skew to that. Like ideally, in a very ideal separation, you have these nice clean Gaussians that are symmetric, but you can also have this peak here is showing tailing. So is this guy here, and fronting, I'll draw this as a dash line here. Fronting would be if you have a skew at faster timescales. Tailing is if you have a skew behind the peak as well. So these are like the bane of chromatographers' existence, of fronting and tailing and decreasing that resolution. So even with this term here, these very I guess they are quantitative, but like empirically based descriptions, you're still missing a lot of information with these basic definitions. So there could be more complex descriptions for these empirical equations as well. And I also want to mention that there's terms that come from distillation theory. Again, from like the 1950s or so, 1930s even, that are applied to chromatography. So distillation is when you're separating things based on their boiling points as well. And this terminology for the chemical engineers here would be able to explain this in more detail, but these are applied in chromatography as well. So these are plate height, which tries to describe the variance per unit length of the peak. So this is going to be, it's abbreviated as H, and this is going to be the width of your peak squared over the position of that peak. And this has some hand-wavy connections to some physical processes that the most common equation that you'll see used to describe Van Diem, okay, that one's fun. The Van Deemter equation, where this plate height is then related to this equation A plus B over U plus C times U, where A is related to the flow pass, B is related to the diffusion, and C is related to the mass transfer, and this U is the flow rate. So mass transfer is the equilibration between the mobile phase and the stationary phase for your analytes. B is the diffusion pass that takes, or the flow that would be the diffusion constant as it's going through the column, and the flow pass have to do with the porosity of the particle. So this Van Deemter equation that was developed in the 1950s is trying to relate the performance to some physical values of the column, but these values are literally just constants that, like, okay, if we want to get down to that smaller scale or physical properties of the column, you need a better description than just a constant there. And then finally, this plate height is then related to the number of plates, or N. And that has to do with how efficient your column is. And N is going to be the length of your column over the plate height, which then can be related to 16 times your retention time over the width of your peak squared for those terms. So I'm ranting about like these terms from distillation theory because the number of plates is this term from distillation theory, and it's not a physical thing. They're called hypothetical plates. And that has to do with like, you literally just assign this number, and it has to do with like, they're saying like how many zones it's passing through, how many plates it's passing through. The smaller the number of N, like the faster your separation is. But the fact that it's like literally termed hypothetical just bugs me. So anyways, some equations that you should be aware of. I'm kind of knocking them, but you should be aware of these if you're doing chromatography and the like. So any questions about this? Okay. So with the time we have left, I'm going to put you guys in some breakout groups. And let me see. And I'd like you guys to, we talked about all the different types of stationary phases, and based on these different conditions, group one has a more straightforward one that you might even have in your house for a water softener system, but also some examples that I found through literature as well of what type of stationary phase or method would you be using for these separations. And also remember, it could be electrophoresis or a chromatography method. So I'm going to stop the share if you guys want to grab a screen grab, or I'll start sharing this after I put you guys in breakout groups.